Okay, so today we're very excited to have Julia Kreiner. She's a 2021 Early Career Scientist awardee for the ESS. She is a PhD student at the University of Toronto, which I just learned she just finished a few days ago. And so she'll be taking a postdoc position at the University of British Columbia. And today she'll be talking about the evolution of herbicide resistance. And so thank you very much, Julia, for joining us today, and I'll hand it off to you.
Yeah, thanks so much for giving me the opportunity to speak to you guys. I'm really excited. I've already had some really interesting meetings, so looking forward to chatting with you more. Here, I'm just gonna screen share. And you're seeing just the slide? Okay, perfect. Awesome. Yeah, so today I'm going to tell you about the work I've been doing for my PhD thesis at the University of Toronto, and I've been working with Stephen Wright and John Stinchcombs to understand broadly the population genomics of herbicide resistance. And the story I'm going to tell you today is work I've done to characterize the repeatability and the genomic architecture of herbicide resistance evolution. So as ecologists and evolutionary biologists, I think a lot of us are interested in the question of when populations can adapt to extreme environmental changes and actually trying to predict this in the wild. And we might be interested in that with respect to slow gradual changes or relatively slow gradual changes like climate change, or maybe even really rapid selective pressures like the application of herbicides. And one really great framework for thinking about this question or for addressing it is evolutionary rescue theory. And the reason I like this is because it takes into account not just demographic history, so the role of past demographic processes and current population size for the likelihood of undergoing evolutionary rescue or local adaptation, but also whether there's sufficient genetic variation to draw from, whether that's new mutation or standing genetic variation. And so at one extreme end of this continuum, populations might undergo local extinction if their populations are too small, or if there's not enough genetic variation, whether that's new or pre-existing. But on the other hand, populations might persist with very little perturbation to their population size, and understanding what processes facilitate this is really exciting feat of evolution, our studies of evolution. And I think one of the most kind of impressive examples of evolution or rapid evolution or evolutionary rescue in the wild is the rise of herbicide-resistant weed populations. And so the number of herbicide-resistant weed species has rapidly increased over the last 50 years, the last half century since herbicides were first introduced in the early 1950s, especially so for acetylactase synthase inhibiting herbicides. So this is one chemical class of herbicides known to target this gene, ALS, that produces amino acids. But another one that I'll be focusing on today, additionally, is resistance to glyphosate herbicides or Roundup ready herbicides. This is a much more recently introduced or invented herbicide, but already we're seeing quite an incidence of the evolution of resistance in weed populations. And so this is a really remarkable example of repeated evolution, and it's clear it's occurred a lot among species. But what's less clear is how often this is arising repeatedly within species and even within populations, and what the underlying genetic variation is that leads to these patterns we're observing. And so for the evolution of herbicide resistance, there's really two genetic bases that we like to talk about. As evolutionary biologists, I think we'd classify this as monogenic or polygenic adaptation, but weed scientists, we talk about this as target site resistance or non-target site resistance. And the idea being is that particular single nucleotide polymorphisms very commonly seem to confer resistance by changing the conformational binding site of the enzyme for which the herbicide is designed to inhibit. And so in doing so, that enzyme is no longer, suppressed by these herbicides, and they can still produce those necessary amino acids. But on the other hand, there's a variety of different mechanisms which are much less well understood from a genetic perspective for how or what changes lead to these resistant mechanisms. And so some of these mechanisms are reduced penetration of the herbicide, altered translocation, enhanced metabolism, et cetera. But it's really been a black box for understanding kind of the underlying genetic basis of resistance and how widespread this is across populations. So when we're thinking about the source of these kind of genetic architectures of resistance, there's really these three evolutionary scenarios for the spread of herbicide resistance. And of course, these also apply to other types of adaptive traits. And the first one is that resistance is rising through new local mutations. And a lot of my work has really been inspired from the literature that's been developed for understanding adaptation to antibiotics, especially and antivirals, especially HIV resistance. And there's some really cool, even like footage and papers showing how rapidly this evolution can occur in real time. But also increasingly, we're kind of recognizing the importance of selection on pre-existing variation. And there's really cool example like the three-spine stickleback, loss of body armor, and their transition between salt and freshwater environments. But lastly, it might not be through these new local mutations or standing variation per se, but we might actually see that resistance is spreading across the landscape through gene flow. So these adaptive or these high fitness genotypes are selected to colonize new environments. And I think it's been pretty breathtaking and remarkable seeing us actually be able to track the spread of COVID variants globally and through genomic epidemiology. And so one day we might be able to take similar approaches to understand the spread of resistance across the landscape. And that's the type of work that I'm going to show you some tastes of today. So we can actually characterize these three different origins using genomic signals. And so I'm going to just walk you through what some of those patterns look like. So time here along the x-axis, frequency of an allele on the y-axis, and you might have some mutations arising at a particular locus, but before that an environmental change occurs, those mutations are in favor, so they're neutral, they arise, but then they're lost. When an environmental changes occur, such that now that mutation might actually lead to an adaptive benefit, when it arises, it will rapidly increase in frequency in the population, such that it spreads onto many different haplotypes. So now that beneficial mutations, it's at a very high frequency, and it brings along with it the linked neutral genetic variation on the haplotype on which it arose. So all of these linked neutral alleles are fixed along with that beneficial mutation. And so this leads to an increase not only in the frequency of allele, but the homozygosity for that haplotype or the homozygosity around that selected locus. Together, this results in a reduction in diversity around the selected site. So this is something you might expect. If there was a single origin of a target site resistance allele, we would see this hard selective sweep patterns. And we can infer this was probably likely to be driven by selection on a new mutation, at least new in the context of it arose after the environmental change. But we can actually use these patterns also to say something about the age of that mutation. And so if that allele arose before that environmental change occurred, it might not have been selected so strongly, right? It wasn't as favored, at least as much in that previous environment. So it has time to kind of float around at low frequency in the population, recombine onto different genetic backgrounds. And now when that allele increases in frequency, it's going to bring with it multiple different haplotypes. So you don't quite get the same reduction in diversity and the same reduction in heterozygosity. So it's a softer selective sweep signal. And you can also get this softer signal if you actually get multiple origins of these adaptive mutations, even if they occur after the onset of the environmental change, because now there'll be these multiple haplotypes for which these different beneficial alleles arose on, spreading through the population. So we can use these patterns to infer something about the evolutionary history of these adaptive mutations. And so for today's talk, I'm going to be using these approaches and thinking about ways that we can test kind of the genetic basis of evolutionary rescue to herbicide resistance in a few different ways. The first way, I'm going to be characterizing the adaptive origins of a complex monogenic resistance polymorphism that's actually a gene amplification, so a little bit more complicated than some of those expectations we would make for a single nucleotide polymorphism. But then I'll bring it back to these really well-characterized single nucleotide polymorphisms conferring resistance and show how we can use population genomic methods to really powerfully infer the number of origins and their evolutionary history. And then lastly, I'm going to show you the work I've done to characterize the polygenetic basis of glyphosate resistance and try and resolve the importance of this genetic architecture compared to much more the target site resistance mechanisms, which are much more easily assayable. And therefore, we kind of think that it's kind of the main mechanism of resistance. And if I have time, I'm excited to tell you a little bit about the work I'm planning to continue to do for my postdoc. So throughout my PhD, I've been working on developing Amaranthus tuberculatus as a system, a population genomics model system. And it's been a lot of fun, and I've been really lucky to get involved with this collaboration. And I'll tell you a little bit about my collaborators in a couple slides. But it's a really interesting system for understanding herbicide resistance because it's one of the most problematic agricultural weed species in North America. There's been single individuals characterized with cross resistance to up to 6 different chemical modes of action. So highly resistant plant populations to the extent to which weed management strategies are no longer efficient or effective. It's also dioecious, which is pretty rare for especially an annual diploid species, so meaning it has separate sexes. And while it's been problematic in North America, especially the Midwestern United States for at least the last 50 years, it's only very recently become problematic for agricultural practices where I am in Ontario over the last decade. So we really wanted to first understand how herbicide resistance was spreading, but also where these populations, these new agricultural populations in Ontario are coming from. So this species has a pretty interesting demographic history. There's actually 2 subspecies with partially overlapping ranges. So to the northeast, there's this riparian subspecies, var tuberculatus, which actually follows the Mississippi and Missouri water basin. So it's very riparian, found in these natural habitats. But to the southwest, var rudis is much more associated with disturbed agricultural environments. And some of the first genetic work on the species used microsatellite data to understand the distribution of these 2 varieties across the range and how that might be affected by agricultural environments. And so they actually, when they allowed for 2 ancestral populations and they asked how those ancestral populations would be distributed across the range, they actually found this expected decline in ancestry that recapitulates this hypothesis actually from herbarium specimens of these range limits. And actually it led them to hypothesize that it might have been the expansion of this var rudis western type that led to much of the agricultural invasion we're experiencing today, because there was an overrepresentation of that ancestry in these agricultural environments. So for some of the first work from my PhD, I wanted to kind of do this population genomic test of evolutionary rescue theory and think about how the adaptive origins of herbicide resistance might relate to population history, especially in the context of these 2 subspecies that seem to have been evolved in different environments for these different timescales. So the first question being, what are the demographic origins of agricultural populations? And then with that backdrop, allowing us to better understand how many genetic origins of glyphosate resistance there are and from what source they arise. So in order to do this, we had to develop genomic resources for the species, and this was done in collaboration with Detlef Feigl, who helped us with a lot of the sequencing effort. Patrick Turnell, who's a weed scientist at the University of Illinois, and Peter Sikkema, who's a local Ontario weed scientist who is helping us track the spread of these populations into Ontario. So this was a really highly collaborative effort. So we were able to produce a high quality PacBio de novo assembly with almost with scaffold resolved or chromosomal resolved scaffolds. And we resequenced and phenotyped for glyphosate resistance or Roundup resistance almost 180 individuals from 19 agricultural and 3 natural populations. So what I'm showing you here in these pie charts is just the proportion of individuals in dark resistant to glyphosate herbicides. And so we have collections from Missouri and a bunch from Illinois where the invasion is very problematic and escaping weed management. But I also want to bring your attention here to Ontario, and especially to Walpole Island in Essex County. And these are actually some of the only locales for which agriculturally amaranthus was identified in Canada. So we collected these populations to understand where they came from. And for good measure, and as this like kind of nice natural comparison, I found some natural populations nearby. So we could kind of disentangle whether or not these have recently evolved from local natural populations, or maybe they spread from the United States. And these collections were done, of course, in the context of the historical range of these two subspecies, spanning the allopatric and sympatric parts of the range. And so for the first results, I really wanted to see how well genome-wide data reflected that climb we were seeing from the microsatellite data. And what we saw is to the west, our Missouri population was homogeneous for putative var rudis ancestry. To our most eastern population, homogeneous for var tuberculatus. This is what we expected to see. Let's see if I can go back here. Illinois, which is actually a region of symmetry between these two historical ranges, is admixed. About 13% of the genome introgressed from this eastern var tuberculatus type, but predominantly var rudis. But what was really interesting is when we jumped over to Essex and Walpole, these two new regions in Ontario, we see very different demographic origins of these agricultural populations. And actually, Essex seems to be homogeneous for this very Western var rudis ancestry. And it's important to remember this is actually outside of the historical range of var rudis. So it seems as though Essex seems to be a putative seed translocation event, but just nearby, less than 100 kilometers away, Walpole Island actually is predominantly our local kind of var tuberculatus ancestry. However, with a considerable amount of introgression, almost 23% of the genome introgressed from this Western type. And so when we're coming back to the question about what the demographic origins of these agricultural populations are, it seems as though there's been multiple origins, at least for these new populations in Ontario, that seems to be putatively related to the expansion of var rudis, like was hypothesized, in that Essex seems to be the seed translocation event of Western var rudis ancestry. But very different origins in Walpole, seemingly, de novo adaptation to these agricultural environments on this var tuberculatus background, which is not typically found in these disturbed agricultural environments. So with that understanding of the demographic history of these populations, I next wanted to look at actually the genetic origins of glyphosate resistance and what source they arose from in terms of timescale, whether that's repeated new mutations or through gene flow. And so I've talked about target site resistance and mostly how single nucleotide polymorphisms can lead to resistance, but you can also have target site resistance in terms of this monogenic architecture occur through the amplification of the target gene. So the gene targeted by glyphosate herbicides is actually EPSPS. And coming into this, it was actually remarkable that amaranthus had been reported to be one of the only species that evolved resistance to glyphosate through the amplification of this gene. And so when I looked at our copy number kind of distribution in resistant individuals and susceptible individuals in our population, and if I just correlate copy number against resistance, we found that this amplification of EPSPS was actually the key mechanism of glyphosate resistance in our populations. We even found individuals with up to 30 copies of the EPSPS gene, and these of course were highly resistant. And so in Walpole, it actually explained almost 50% of the variation in resistance, but much less in Essex in the Midwest, implying there are actually other mechanisms going on here. But I'm just going to be focusing on, for this chapter at least, or this part of the talk, this amplification. So we can actually go back to those genomic signals I said we can use to survey kind of the time scale and the number of origins of this mutation. In particular, we can look at the reduction in diversity, the allele frequency differentiation, and the excessive homozygosity in our resistant compared to our susceptible individuals. And we can see if we have these signatures of selection, which we know there should be selection occurring on these sites. And so if there's differences going on here, that might tell us something about the number of origins. So when we look across the chromosome that contains the EPSPS gene, here I'm showing that by the vertical dashed line, we actually see these hard selective sweep signals in Walpole, where we see these strong reductions in diversity and excess homozygosity associated with these individuals that are resistant at this gene. And this was really remarkable, seems to be evidence for selection on this de novo mutation that occurred very recently, but we don't see that same pattern in these other agricultural regions that harbor lots of glyphosate resistance, and they harbor lots of this gene amplification, so Midwest and Essex. And so we wanted to see if we could look at this another way, a more direct way that actually quantifies similarity in the amplification profile, or the number of gene copies, that can give us more direct information on the number of origins, as we expected that might be underlying the difference in the pattern we were seeing. So what I'm showing you here are those copy number profiles. And so here I'm just showing you each of these kind of rows is an individual, or an individual's haplotype, and the copy number of a loci around the EPSPS target gene. And so you can see some individuals have extremely high copy number in this region. This one up on average is 30 times the genome-wide coverage or copy number. And while the individuals vary in their amplitude, it almost looks as though they have very similar peaks and valleys of their copy number, implying it might have been originated from the same amplification. Well, we can actually quantitatively qualify that much more explicitly than just looking at these plots. We can use the Spearmint ring correlation coefficient to quantify the similarity in the peaks and valleys of this copy number profile. And so when we do that for all individuals that harbor the amplification in Walpole Island, this new, var tuberculatus type kind of naive population in Ontario, individuals are almost perfectly correlated for the amplification that they have, implying that, again, that there's been a single origin of glyphosate resistance. And this kind of fits with the expectation from that hard selective sweep signal we were seeing. But it was the other regions where we weren't seeing that hard sweep signal. And indeed, when we looked within amplified individuals in Essex, so within this square, it's showing these within Essex correlations, we actually see very low within region correlations in the similarity of their amplification profile, implying that the amplifications harbored within Essex are actually very distinct. And actually, when we look among populations, especially with correlations from the Midwestern United States, it actually looks as though these Essex individuals are a collection of Midwestern type amplification haplotypes. And that fits with the knowledge we have from that demographic story that Essex seems to be this seed translocation event with very Western Varrotus ancestry you wouldn't expect in this region. And so in the Midwest, that we really think is the epicenter of this kind of agricultural invasion, it actually appears as though there's been multiple origins of this amplification event, where there's high within population correlations, but relatively low among population correlations. Although with some, although it looks as though there's some exceptions to that, with a few high within between population correlations. So this was a really cool way to visualize kind of this complex mutation and the number of origins among it, the number of origins for this amplification among populations. And in Walpole, it seems as though it's, again, adaptation from this new mutation, but very different when we turn to this Varrotus ancestry that we think has been evolving in agricultural populations for much longer times, where Essex seems to be this case of adaptation from gene flow from the Midwest, and Midwest seems to have evolved glyphosate resistance through recurrent mutation. So when I'm thinking about how the adaptive origins relate to population history, it does seem like there's this clear dichotomy where we're seeing soft sweeps in gene flow associated with the recently expanded Varrotus ancestry, whereas hard sweeps and kind of maybe evolution in this mutation limited context in populations that harbor this var tuberculatus ancestry that seems to be much more naive to agricultural selective pressures. So for this talk, I've kind of focused on how population structure relates to the availability of adaptive variation. And next I want to take a step back and actually think about how we can use some of the amazing information that we scientists have curated on characterizing simple single nucleotide polymorphisms that confer target site resistance, and how we can use that information with genomic data to actually get really fine scale inference on the genealogical history of these mutations in a much more powerful way than we could with this amplification, because it really just violates so many of the assumptions that population genetic theory tends to make about rapid adaptation. So for this part of the talk, I'm going to switch gears from glyphosate and talk about resistance to these two different classes of herbicides, acetylactase synthase inhibiting herbicides. Again, this is a gene that produces necessary amino acids, and this PPO inhibiting herbicide. And so there's been a number of mutations characterized, single nucleotide mutations characterized that occur within these genes that lead to resistance at the individual level. And for today, I'm going to be focusing on these two mutations, a non-synonymous mutation codon 574 and 653 in ALS, and also this 210 deletion of the 210 codon at this PPO gene. And the reason I'm focusing on these are particularly common target site mutations in the same samples that I've been talking about. And so if we're interested in understanding the number of origins of these mutations, one approach we can take is to visualize the gene tree of these regions, and not only just visualize, how related individuals are, how much shared evolutionary history they have in this region, but also map on the status of an individual's haplotype for whether or not they harbor that target site resistance allele. And so when I map that on, the 574 status and the 653 status, you can see these clusters of resistant individuals grouping across the tree. And that we don't see that all these individuals cluster together, as you would expect from a single origin of this resistant, implies there's been numerous repeated origins of these target site resistance mutations. And it was also really reassuring to see that often these clusters of resistant haplotypes are associated with these really particularly short terminal branch lengths, which implies that the common ancestor on where this mutation arose really rapidly gave rise to descendants relative to the rest of the tree. And so it's a strong signature of selection. So this was really interesting, and if you were to count up the number of origins from this, I think it would be pretty remarkable. I think we see about 6 for 653 and maybe 8 or 9 for 574, 3 for PPO. And so when I saw that, I was kind of like, wow, that's a lot higher, I think, than some of us might expect. And one thing that I wanted to think about was how recombination might actually influence the patterns I'm inferring here. And that's because recombination drives fine scale variation in coalescent history across chromosomes. And so I'm showing this really cool plot. So this is from some work in Heliconius, how recombination shapes patterns of introgression across the genome. And what they visualize here, all of these different colors represent different gene trees for species within the Heliconius genus. And so when you move across the genome, you see very different coalescent histories in these different regions of a particular chromosome, but even at the fine scale level, like this changes very rapidly as you move across the genome or across the chromosome. And so it's possible that this was influencing some of the patterns that I was looking at. And so one way that I can actually account for that recombination effect is through ancestral recombination graphs. So this is a method developed actually in the 1990s, but I think in the last 5 years or so, there's been a lot of work to make this method efficient for genome-wide level analysis. So tractable for analyzing hundreds of individuals at the genome-wide scale. And so what is an ancestral recombination graph? Well, you can imagine, you can imagine representing tree sequence across the genome. So for a particular locus, you can ask about the extent to which individuals coalesce more recently or further in the past. And the ancestral recombination graph brings these different trees across the chromosome together by representing not only coalescent events backwards in time, but recombination events. So the only thing that distinguishes one gene tree here from the next is that one has experienced a recombination event. One region of the chromosome has experienced a recombination event that makes that gene tree unique. And so by incorporating both coalescent and recombinational history, we can really more precisely infer not only the number of mutational origins, but things like the mutational age and signatures of selection. And this is work I've been collaborating with on with Aaron Stern and George Sandler. So now I'm showing you the tree sequence I've extracted for my particular loci of interest, so those target site resistance loci, those 3 loci, from the ancestral recombination graph that represents both coalescent and recombinational history. And when you count for recombination, you actually find pretty different patterns of clustering. You find 6 origins of the 574 mutation, but only 2 origins of the 653. So if you remember from just the gene tree approach, we saw almost 6 clusters. So it actually appears that recombination has really shaped kind of patterns of haplotype relatedness across this entire gene that would have misled us when we were thinking about characterizing the number of repeated origins. Even for 574, accounting for recombination brings us down to about 6 origins as opposed to 8. So not only does this show extreme mutational parallelism for these target site resistance mutations, but also a role of recombination, and that these mutations actually seem to be old enough in that recombination has played a role in their evolutionary history. So I've characterized like 11 origins of these 3 types of target site resistance mutations, and we can actually map these on to our geographic populations to understand something about how local these mutational origins are, but on the other hand, the role of gene flow in determining their spread across the region. And largely, you don't find a ton of really local signals of high frequency mutations confined to a single population. There's one example, the 653.7 mutation, which is actually one of the largest kind of selective sweep signals we see, is pretty localized to Ontario. It's at really high frequency in Essex County and somewhat in Walpole Island. And it's not found in Illinois, but it is found in one region just outside of Kansas City. So even this kind of highly localized mutation is still found in the Midwestern United States. But otherwise, just, by glossing over this, you can pretty clearly see that most mutational origins are found in many different populations. And conversely, most populations harbor many different mutational origins. So really a widespread role of not just repeated origins of resistance, but gene flow in their spread across the region. We can actually also use these gene trees to infer something about the likelihood that selection has occurred consistently over the course of that mutation's evolutionary history since that mutation first arose, or whether evidence for selection has varied across it. So whether selection seems to be likely occurred over a more recent timescale than when that mutation first arose, and that's consistent with selection on a standing genetic variation. And in particular, what we can ask about is the probability that the observed skew in the number of descendants of a focal lineage is particularly extreme given the rest of the tree over those 2 different timescales, right? And that's the pattern that I pointed out here. You see the very, very short terminal branches implying a particularly extreme skew associated with that resistance lineage. And so we can do that over these 2 timescales and estimate, the probability that selection has occurred in these 2 different scenarios. And what we find is evidence for adaptation from both standing variation and de novo mutation, but it varies, and it varies considerably. Of these 11 mutations, only 5 of which show significant signals of selection. So what I'm showing you on the Y, I should say, is the negative log 10 p-value of these tests for each independent origin. And so 4 of these origins show significant evidence for selection over the entire course of that mutation's history, on selection on a de novo mutation, but 2 of which show selection on standing variation. And it varies, even within a particular type of mutation. So here, the ALS 574 mutation, you see both types of selection. for the 653, you see selection for one origin, but not the other. And so it's clear that there's a lot going on here, not just probably temporally fluctuating selection like I'm showing you here, but I'm sure that there's tons of geographic fluctuating selection based on variation in herbicide use that might lead to some of that heterogeneity we're characterizing here. So not only have we characterized extreme parallelism in mutational origins, but I'd say also heterogeneity in their success, in that it seemed as though, not all target site resistance mutations show strong evidence of selection, presumably in large part due to the selective environment in which they arose, whether herbicides had been applied repeatedly in that time span. But also we found a widespread role of gene flow in the spread of these resistance mutations, and actually a maybe underappreciated importance of recombination in shaping these target site resistance in patterns of similarity of these target site resistance mutations. So for the last kind of meaty part of my talk, I'm going to switch gears from thinking about characterizing repeatability of target site resistance to just characterizing the polygenic basis of glyphosate resistance, so resistance to Roundup herbicides. And like I alluded to before, because these target site resistance mutations have been so well characterized, we can actually easily go into populations and assay the frequency of these mutations. And I've done that to an extent by just going into the genome in my previous section and just assaying the frequency of these known target site resistance mutations. So it allows us to really easily kind of evaluate how widespread these mechanisms are, but we don't really have an equivalent understanding for the polygenic basis of herbicide resistance. So for this, I really wanted to think more broadly about how selection from herbicides actually shapes genome-wide patterns of diversity, not just kind of these hard selective sweep signals that are really localized to these regions, but how widespread are these signals of selection across the genome to herbicides. And for me, that meant characterizing the polygenic basis of non-Targetsite glyphosate resistance. Well, what does that mean? Well, I'm interested in the number and the distribution across the genome of genes that correlate with glyphosate resistance, their effect sizes. So how large effect are these genes, or do they vary a lot in how much resistance they actually confer? And how frequent are these genes, or how frequent are these alleles across populations? What are the associated signals of selection, and how important are these resistant mechanisms, this polygenic resistance, relative to this more easily assayable monogenic resistance? So from the first part of my talk, I was working a lot characterizing kind of the origins of EPSPS copy number amplification. And I said that it explained a very large portion of the variation in glyphosate resistance. But I can actually show you here, that another way. And so what I'm plotting here are another known mechanism of glyphosate resistance, this EPSPS proline to serine substitution at codon 106. So you can see that's at a particularly high frequency in a couple populations. There's some individuals that have both of these target site EPSPS mechanisms, the amplification and the SNP. But there's some individuals that are resistant, but show no known target site resistant mechanism. And those individuals are in this bright green. And so you can see there's actually a considerable number here that seem to, that seems to imply polygenic mechanisms in conferring resistance. And so not only do we have these individuals with only non-target site resistant mechanisms at the population level, if we regress resistance on these two types of mechanisms, there's still lots of variation. If we regress resistance on these two target site mechanisms, there's still lots of variation unexplained. So it seems as though non-target site resistance actually occurs alone, conferring resistance robustly, but also complementary to these large effect target site mechanisms. So it seems as though we have lots of variation that we can use to try and assay the genetic basis of non-target site resistance. And what some of you might have expected, the way we did this is through a genome-wide association. And so at every locus across the genome, we perform a linear regression asking how much genotypes across individual correlate with resistance levels. And we can do this as a first pass test with no covariates and just say, if we just regress resistance against genotypes genome-wide, do we see the large effect locus that we know is causal for conferring resistance? And indeed, some of the largest peaks were associated with EPSPS and even that EPSPS amplification. And so this was really reassuring to see, okay, GWAS actually works as it should. It's identifying this causal gene. But I really wanted to figure out resistance that was unexplained by these target site resistant mechanisms. And so what we figured we could do is we could actually use EPSPS copy number and this proline 106 to serine substitution as covariates in the GWAS. And so that will remove the variants explained by these target site resistance mechanisms and allow us to explain an independent genetic architecture of resistance. When we do that, we see very different patterns of association. I would say this here is resistance associated here. Here, the second row is really a GWAS for non-target site resistance. And we see no association at the chromosome, very little association at the chromosome that contains EPS, especially at that gene. And you can see how much using these covariates changes your inferred distribution of non-target site resistant alleles across the genome. And so that's really what I want to focus on here, this later GWAS. When we look at what the genes actually are that are significantly associated with non-target site resistance, we see almost 573 alleles across almost 250 genes across the genome associated with non-target site resistance. And they actually show, kind of shockingly or excitingly, enrichment for relevant functions for herbicide resistance. The most significant being response to chemicals, response to stimulus, but a lot of metabolic processes. And this is what we would expect. EPSPS is a gene involved in the production of aromatic amino acids. And a lot of the processes we see are related to metabolic processes up or downstream of that shikimate pathway. So this seems as though it kind of implies there's been selection on these different functions, but on the other hand, we also saw enrichment for these less kind of directly related resistant categories, multicellular organismal development, multicellular organismal processes, and these are genes that function in things like reproduction, circadian rhythm, and photomorphogenesis. And so while it seems as though there might be direct selection on these genes, there might also be pleiotropic constraints acting on the genetic to shape the genetic architecture of resistance. And so I wanted to see how extreme the patterns of selection or constraint actually are on these genes. And so we had to come up with an expectation. How much selection would we expect to see for the frequency of resistance we see and the population structure in our samples, and the population structure being a really important caveat here, because we know there's some issues with GWAS that even controlling for relatedness with a kinship matrix might not do a great job. And so what we decided we could do is we could actually come up with a null that accounts for the fact that our populations vary in their population structure, but they also vary in their architecture of resistance, just by the fact that you can see the frequency of non-target site resistance is variable across these populations. So worst case scenario, if population structure was really highly correlated with genetic architecture of resistance, you would have a really hard time finding true positives, true alleles that correlate with resistance that aren't driven by population structure. So we came up with this permutation approach where we randomize phenotypic assignment, resistance assignment across individuals within agricultural regions and within populations. And what that does is it retains the association between population structure and differences in genetic architecture of resistance, and gives us a null for what type of patterns of these alleles we would expect with respect to their frequency, their effect size, given the structure of our data. So the first thing I wanted to look at was the effect size of these alleles and their frequency. And the effect size, I think, tells us something about selection along with the allele frequency. And what we found was, compared to these null expectations, we found particularly large effect alleles. but also that those alleles were at particularly low frequency. And I think this speaks to both the role of selection and constraint, in that it's drawing on these large effect alleles, but these large effect alleles are kept at low frequencies, implying that they might have pleiotropic costs in the absence of herbicides. Another way we can look at signals of selection are actually through these haplotype level population genomic analyses. And so what I'm showing you here is this thing called the integrative haplotype homozygosity, and that measures the extent to which homozygosity is enriched in these resistant haplotypes, not only at a particular locus, but the length of that homozygous haplotype. And so we can see that resistance is associated with particularly long homozygous haplotypes compared to susceptible haplotypes. And that's the case even when we standardize by allele frequency. So even though these alleles are rare, given the rarity of these alleles, we still see much more homozygous and longer homozygous haplotypes associated resistance alleles than we would expect even against these pretty conservative nulls that we've come up with. So again, evidence for partial sweeps of these polygenic resistance alleles. The last thing I wanted to look at was the importance of these polygenic mechanisms compared to these monogenic target site mutations. And I think we can interpret this in the context of almost 80% of the individuals included in this study harbor at least one target site resistance mutation. So we already know that target site resistance, these large effect mutations, are very widespread. And when we regress resistance on copy number and the SNP target site resistance mutation, almost 33% of the variation in resistance is explained by these target site mechanisms. In comparison, on the background of this the genomic background of these target site resistance mutations, from this GWAS approach, we can actually see that almost 25% of the variation in resistance is explained by polygenic mechanisms. So really comparable importance of this polygenic mechanism compared to target site. So I really think it speaks to the underappreciated importance of polygenic resistance. It was really fun to think about how we can use genetic covariates to uncover cryptic genomic architecture, that even for polygenic resistance, we're seeing strong signatures of selection, but also pleiotropic constraints, and that these resistance alleles have been kept rare and that they have effects on growth and reproduction. So I've talked to you a lot about the repeatability and genomic architecture of herbicide resistance evolution today, from the effects of population structure on the availability of adaptive mutation and the role of new mutation in standing genetic variation. And it's been fun to think about this in the context of evolutionary rescue, but also apply some of these theoretical expectations for what selection should look like across the genome to infer something about the repeatability of these mutations and how that might differ across these different genomic architectures. And so in thinking about, herbicide resistance evolution in amaranthus and the role of standing genetic variation, new mutation, and gene flow for its spread, it really seems like it's all of the above. This species seems like it's going to be a real issue for management going forward, and that we have to think about all of these different mechanisms of the spread of herbicide resistance to really get it under control. But we saw that it does depend to some extent on population history, and that these naive populations seem to be mutation limited, evolving through new mutation predominantly, but it depends again on the genetic architecture. I just want to take a quick second to tell you a little bit about the work I'm planning to continue on throughout my postdoc and grow kind of on this work I've been doing in my PhD, and that's thinking more broadly about the basis of contemporary agricultural adaptation and seeing if we can actually quantify temporal change in agriculturally fit alleles. Though I've been lucky enough to have a chance to get out in the field and really observe these plants, and it's been remarkable to me to just observe some of the variation we see in these natural and agricultural populations. So this individual here in a fallow field, agricultural field, is like 10 feet tall, just towering over the landscape. And you can see here another amaranthus individual really outcompeting soy for resources. And so in seeing these differences, I really wanted to ask about what underlies agricultural adaptation, the phenotypes and genetic loci, the extent that gene flow between these two subspecies has facilitated or hampered the agricultural invasion of weedy water hemp, and how the underlying genetic alleles, how the frequency of these alleles have changed through time and with climate. So towards this goal, I've actually performed pairwise sampling of natural and agricultural populations across the range. This allows us to disentangle kind of these fine-scale environmental changes from the background of of this kind of complex demographic history and geographic effects. But along with these contemporary samples, I'm super excited to have curated almost 100 historical herbarium samples that span the same geographic range, but date back to 1800, so that we can, once we have a better understanding of the phenotypic and genetic differentiation among populations, we can track in real time how those alleles have changed through time. And this was really fun work. I've already started, actually I have sequence data, but I worked with Hernan Bourbono and Detlef Weigel in Tübingen, Germany, using their ancient DNA-specific wet lab protocols for the extraction of these herbarium DNA from these herbarium specimens. And just to leave you guys with a little bit of a teaser, how the frequency of agriculturally fit alleles change through time and with climate. Well, one of the first things I looked at was the frequency of target site resistance in these samples. And this ALS 574 mutation, one of the most common ones I saw in my contemporary data at almost a frequency of 50%, actually found standing genetic variation for this mutation to homozygous individuals in natural populations that actually predate the onset of herbicides. So 1833 and 1876, while herbicides were only introduced in the 1950s. So clear standing genetic variation for an agriculturally fit allele, and the frequency of which has changed really dramatically over the last 200 years. So with that, I just want to thank you guys so much for having me. I'm super excited to be here and to chat with you all. And I want to thank my labs, Stephen Wright and John Stinchcombe, the U of T greenhouse staff, all the undergraduate research support I've gotten, my collaborating labs, especially Detlef Weigel, Patrick Trinell, Peter Sikkema, and the co-authors on this research. And yeah, with that, I'd love to take any questions.
Thank you, Julia. Feel free, we have a few minutes for questions. Feel free to put them in the chat or speak up. And I'm going to stop recording.